today's haiku. Epithelium and connective tissue form the integument. Very simple, practical haiku today. All right. So, the human body is a really incredible community. It's an incredible community. There's, maybe 8 billion humans on Earth of, approximately 200 different nationalities. And we're constantly at each other's throats and not able to, get along. The 50 trillion cells in your bodies are able to happily coexist in the entity of a single organism. We have 200 different cell types here. I think I was talking to you yesterday, maybe I mentioned there's this really interesting project going on right now where they are taking tissue samples from humans and subjecting them to scrutiny of a sort that has not happened before, and they're actually identifying new cell types in an ongoing basis here. The woman I was listening to her talk, the woman who's heading up the project believes that they could identify as many as 30 or 40 new cell types that have not been, when they're completed with the project. Cells that are in very low copy number, but serve unique purposes in the body. So this is, to say 50 trillion cells and 200 different cell types, to have a round figure like that is appropriate because I don't think they have the exact number. And yet it's not all the questions are resolved. So these 200 different types of cells collect themselves in different ways to make the variety of tissues that are in our bodies. What is a tissue? Well, it's just a certain aggregate of a number of these cell types and their cellular products. And they're going to form a tissue continuum that's going to have a very specific and limited function in the body. These tissues then can make up in various forms the organs that we are very familiar with, such as the kidney or the stomach, etc. So what is an organ? It's a structure that has discrete boundaries. You can say, the stomach has this shape, and it has very discrete functions. It has more than two or more tissue types. It's not a single tissue. So a single tissue cannot form an organ. There are four basic tissue categories, and they can be divided into the non-excitable tissues, meaning epithelial and connective tissue. This doesn't mean that they're not exciting, they're just not excitable. And then I know that was bad, neural tissue and muscle tissue are the excitable tissues. So we're going to cover epithelium and connective tissue today, and then tomorrow I'm going to set the foundation. I'm going to have a lecture basically on cell physiology that'll set the foundation for our discussions of excitable tissues. All right. So, I guess first to give broad overviews of them. Epithelial tissue. There's two types. There's true epithelium, and then there's glandular epithelium. True epithelium forms the covering over exposed surfaces. So, for example, your skin is, the very outer surface of your skin is an epithelium, or the inside of your nasal passage, or the tubes in your kidneys. Those are epithelium. It's the exposed surface on any structure. All right, so inside of your gut, inside of your ear. These are all epithelium. And then connective tissue is a very descriptive term. It's just tissue that somehow acts as a glue, right? It's going to connect an epithelium to maybe some underlying muscle tissue, or it's going to form fascia, fascial plane between layers of muscle, or it's going to form a sheath around a nerve fiber. Connective tissue also has some subsets that include the blood cells. So there are fluid connective tissues. All of the erythrocytes and leukocytes that make up our immune system are considered to be connective tissue. Fat tissue, adipose tissue, is a form of connective tissue, and the principal job there being to store energy. So we're going to explore just an overview, a survey of both of these in the lecture today. So here's a bit of an outline for the tissue types. In the red box, you see the epithelial tissue, and I've broken it up into the true epithelia. This is the types of epithelial tissue that act as covering over a surface, and then glandular epithelium. In true epithelium, in lab yesterday, we talked about the morphological distinctions, didn't we? We talked about simple versus stratified. That was a single layer versus stacks. And then the shapes, squamous being a squished down, flattened epithelium, the cuboidal, and then along this continuum we have the columnar. Now there are, in these categories, some that have a hard time fitting in very clearly. So for example, I didn't talk about transitional epithelium yesterday. This is sort of a cuboidal, kind of a columnar. It's a type that you would find in a very, very stretchy surface. So for example, inside your urinary bladder. This is a structure that on a regular basis has to expand and then come back to shape. That would be a transitional epithelium. It doesn't really fit this category. And then there's the pseudostratified columnar epithelium. They call it pseudostratified because on histological section it looks like it's stratified, but it's pseudo. It's not. It's just faking. It's because the columns sort of intertwine with one another, and it's a little bit resistant to clear delineation on single 2D sectioning. And then the glandular epithelia. We have endocrine glands. These are glands that are going to release their products, their cellular products. So glands in general are epithelial cells that form aggregates that are these structures called glands. And those cells release their cellular products into the bloodstream if it is an endocrine gland. Whereas an exocrine gland is going to release its cellular product not into the bloodstream. It's going to release it into onto the surface of a duct. So it will release it onto the surface of that epithelium that will form some sort of a duct that is going to communicate with the outside world. All right, and we'll look at those differences a little bit. And then I'll move on to connective tissue, and you can see I'll go through this hierarchy when we get to it. The muscle tissue and neural tissue stuff is there just for completeness, but we'll devote lectures to each of those in their own turn. And then finally, these membrane types. And when I talk about membrane here, I'm not talking about lipid bilayer. I'm not talking about plasma membrane. I'm talking about a tissue level membrane. So aggregates of cells that form an epithelium on top of an underlying connective tissue. There's 4 types of tissue membranes. We talked about one of them yesterday in lab, the serous membranes. That's a type of tissue membrane. We're going to go through each of those 4, some of them in greater detail than others. All right. That's kind of where we're heading. I'm putting together all the the epithelium and connective tissue, so I can actually just talk about tissue membranes, because that's sort of the point. All right. First, to get into details on epithelial epithelium, and I apologize for those of you who've had a little bit of cell physiology in the past, or maybe got this in your biology, but I'm not taking anything for granted, so I'm just going to go through some basic cell biology with you. As I said, epithelia covers surfaces, as opposed to the other category, glandular epithelia, which is going to produce secretions. All of these epithelial cells are going to have certain characteristics. So epithelium has particular characteristics that we can identify regardless of where those epithelial cells are found. The first is that there is a cellularity to them. And what does that mean? I thought that like all cells are cells, right? What does that mean? Cellularity here means that there are discrete cellular junctions between one cell and the adjacent cell. All right, so there are junctions between these cells. And I'm going to, in another slide, I'm going to open that idea up a little bit and talk specifically about the proteins that make that happen and the implications, et cetera, et cetera. But there's a certain cellularity to it, which enables that epithelium to form the open sheets that we see in the body. And importantly, there is a polarity. There's a polarity. When I was in graduate school, longer ago than I like to think, but not so long ago, how cells establish a polarity, establish an endedness, if you want to think, was an ongoing area of research. It's not a long settled concept as to how cells actually do this, but it's an important question because initially, embryonically, how does a cell know which end is which? Right? When all ends are the same, how do you establish a polarity to a cell? Epithelial cells are a cell type where this is an important question. So, for example, let's look at this cartoon here of an epithelial cell. We have two ends. In fact, there are two poles, let's say. One is called the apical surface. And in, maps that you look at, we sort of live in this northern hemisphere-centric world where maps always have north on the top and south on the bottom of the map. And in the same way, it doesn't have to be that way, the planet does. In the same way, these cells are usually drawn with the apical surface at the top, right? And on the other end is the basolateral surface. And there is both a functional and morphologic distinction between the two of them. So typically the apical surface of an epithelial cell is the surface that is unencumbered by attachment to anything else. It's open to often the lumen of some compartment like the inside of your gut, or it's open to the outside world, etc. So that's the apical surface. And then the basolateral surface is attached to what's called a basement membrane here. And that is almost exclusively going to be connective tissue of some sort, one of the many types of connective tissue, all right? And this is depicting a simple, this would be, I guess, maybe like a simple columnar epithelium, if I were to guess what this cartoon is representing. In a stratified, of course, the apical surface is getting stacked on top of by other cells, right? But this is a simplistic representation. So then, when you think about the functionality of one of these cells, these epithelial cells, for example, in a glandular epithelium, there's going to be cell products. So this is going to be the nucleus. This is where the genetic material is. You have the endoplasmic reticulum, These are rough endoplasmic reticulum here with the ribosomes depicted in the little dots. That's where the proteins get made, going into a Golgi stack. So the DNA gets transcribed to messenger RNA, which gets translated into proteins that get sent into the Golgi apparatus, and there we go through post-translational modifications, maybe to get clipped or decorated with various glycans or whatever's going to happen, sugars that get stuck on them. They get finished off, right? And they get packed up into these vesicles. Then these vesicles can travel up to that apical surface of the cell and go through exocytosis, the process that I talked about yesterday that calcium was such an important part of. All right. And then there are a number of things that you can see on these apical surfaces. You can have cilia. Cilia are long arms, these like long finger-like projections that can move sometimes in a coordinated fashion or not. They typically are trying to move fluid across the surface. They are increasing the surface area, and they're doing that to be able to increase the potential for absorption. I'm going to tell you right now, because I've sort of struck upon it, I'm going to describe for you a fundamental feature in nature that guides. So yesterday I talked about form and function. I'm going to describe for you a fundamental facet of nature, the universe of physics, I guess, not just life, that guides a lot of the forms that you will see in living systems and otherwise. But so many forms in living systems are dependent upon this idea that I'm about to express that relates to this. So, and that is, you've thought about maximizing surface area to volume maybe before, right? Or, I mean, that's what we're trying to do here, right? We're trying to maximize the amount of surface area that can fit in the limited volume of that interfacial surface. In general, there is a physical principle where you can fit, I'm going to say it in very general terms first, then I'll explain it. You can fit an infinite amount, theoretically, you can fit an infinite amount of n-1 dimensional surface in a finite n-dimensional surface. So what do I mean by that? So for example, if you think about a snowflake has a perimeter that is going potentially towards infinity. It's not, of course, but it in theory it could, right? It's going potentially towards infinity. That perimeter is a one-dimensional surface, right? It's just a line. And so here's our snowflake. I'm just going to make it out of stars. I'll put triangles on the sides and triangles on the sides of that, and triangles on the sides of that, et cetera, if you were to imagine a snowflake like that. This perimeter goes towards infinity, but it's a finite surface area because I can enclose it in a circle of easily calculable surface area. All right? So in this case, n would be equal to 2, a two-dimensional surface. The surface area can contain an infinite amount of n-1. n-1 equals 1. An infinite amount of one-dimensional surface area. Here, we have a finite volume can contain, in theory, an infinite amount of surface area, and so on and so forth. This is why trees have leaves that increase their surface area but fit within a finite volume, the range of the tree. That's why that looks like your lung has similarities to the structure of the intestinal villi in your gut, and so on and so forth and so forth. If you, that like very simple physical reality, if you think about it in general terms, it helps you understand why structures take the form that they do to serve to maximize the efficiency of some certain process. All right, so anyways, cilia, or microvilli, is another much smaller projection coming off the apical surface. The goal is to increase the surface area again, as I had just alluded to. All right, so other characteristics are there's going to be an attachment to a basement membrane. I've already said that. Importantly, and this is an important concept, is there's an avascularity. What do I mean? I mean no blood vessel growth. All right, avascularity to epithelium. And this is actually a consequence of the cellularity, right? There's going to be these tight junctions that zip the epithelial cells together and are going to preclude the ability of capillaries to go through that tissue, all right? So there's an avascularity to epithelium. For example, on your skin, if you were to scratch your skin, if you scratch it not so hard, you just sort of see a white line where you have dug into the epithelium. If you keep going, and if you see a little bit of blood, you have passed the epithelium and gotten into the underlying connective tissue where there is capillary, where there are blood vessels. All right, so epithelium is avascular, and then it is all regenerative. So if you destroy some epithelial cells, or if epithelial cells go through apoptosis, the process of programmed controlled cell destruction, they can regenerate themselves. They can be replaced by stem cells and reform that epithelial structure. So these epithelial cells act as a physical barrier to a number of ways. It controls permeability. So, for example, in the gut, the epithelium there decides what gets in to your bloodstream and what does not. Right? Much of what you eat, we all know, does not get absorbed by the body, just passes through our gut. And this is the job of the epithelium to decide which and wherefore. Epithelium can become the substrate for sensory neurons that are going to provide sensation. All right, so our tactile sense and many of our other sensory organs are intimately involved with the epithelium. And then there are all kinds of specialized secretions that the glandular epithelium make throughout the body. All right, pretty basic. So let's go through the cellularity a little bit and talk about a few of the structures that lead to this. So there are really four categories of cell adhesion molecules that we're going to talk about here. First of all, there are the proteins that are responsible for what's called tight junctions. And I think of a tight junction as sort of like a Ziploc seal along the apical margin of epithelium. They're just zipping all of the surfaces together. So if you can imagine a field of those epithelial cells and those boundaries, those margins that you would see looking down from above, all of those boundaries, all of those margins would be zipped up by these proteins here. And it's a little bit like really strong Velcro. It's a little bit like really, really strong Velcro. And it is these tight junctions that serve a number of purposes. First of all, they are responsible for controlling the permeability of that epithelium, right? So if something wants to get past that epithelial layer, it can't go around the cells. It has to go through the cell, right? And that means that if it goes through the cell has control over what it lets in and doesn't let in, right? The cell chooses what it's going to absorb or not. Yes, sir. Oh, plasma membrane. Sorry. Um, I'll come back to the gap junctions in a moment because I find them to be really interesting. Oh, I guess before I move on from the tight junctions as well. So when you look at the structure of, we could have easily a whole lecture or a series of lectures just talking about tight junctions and the proteins that are involved and the underlying dynamics. But these proteins of the tight junction, there's really two parts. There's this the tight junction that forms this, what I was calling the Ziploc seal around the surface, and then below that there's this adhesion belt, this sort of like terminal web that confers a sense of tensegrity, if you're familiar with that term. But it confers the property of tensile strength to that cell. So you can imagine like a drum. No, I guess it's more like a bicycle wheel, perhaps, where the spokes are pulling in towards the center and balancing the tension around that rim. But it would be as if that bicycle wheel was maybe a hexagon or something and attached to other bicycle wheels around it. So this adhesion belt forms a stable meshwork of protein, filamentous proteins throughout the top surface of the plasma membrane. And there's a lot of other implications that I'm kind of glossing over with that, but it's involved in facilitating exocytosis and endocytosis and communication within the cell, et cetera, et cetera. But it's amongst the things it's doing, it's helping to give that maintain that cell shape and the general tensile strength across the entire sheet of the epithelium across many adjacent cells. So moving on, the next concept we'll talk about is these desmosomes. And I'm sorry, I probably need to climb in that window there and adjust the projector so it's focused better on the screen here. But desmosomes are, when I describe these things, I think back to when I was in high school. I took a shop class, and in shop class there was this thing called the spot welder, and it was this machine that you take two pieces of sheet metal and you stick it in this thing, and these two little fingers would come down and run 10,000 volts, like zap, and it would weld the two sheets of metal together right at that little spot. That's kind of what a desmosome is. It's just a little spot weld between two adjacent cells, and you can see these patches of them all throughout the lateral walls of the cells. They're just these little spot welds, these desmosomes, that, again, hold those cells in place so they're not like sliding around one another when an epithelium goes through some sort of mechanical stress, because all epithelium is going to go through some kind of mechanical stress at some point. Right? Stuff is passing by them, through them, your body's moving, et cetera, et cetera. So these desmosomes, they're little, the whole membrane doesn't need to be adhered to one another, but these little spot welds between two adjacent membranes. And then these desmosomes are connected to a dense meshwork of what are called intermediate filaments. These, again, are filamentous proteins, long filamentous proteins that extend out into the cell and create this spider web of attachments in the cell that helps the cell monitor shape and helps cellular trafficking, et cetera, et cetera. And then, on the other hand, we have what are called Hemi Desmosomes. So the word hemi just means half. A Hemi Desmosome is a desmosome that's attached to the basement membrane. We call it half because there's not another desmosome coming up to meet it from an adjacent plasma membrane, because there is no adjacent plasma membrane. It's just the connective tissue below it. So a Hemi Desmosome is completely analogous to the desmosomes, except that it sends these special proteins that act as sort of anchors or toggle bolts, if you will, that stick down into the connective tissue. Yes, sir, Miles.
like I said, the spot desmosomes are just localized areas. Adhesion belts look very similarly rendered. Are adhesion belts basically?
It's going to be a different set of proteins. There's a different set of proteins for sure. And I think, so a desmosome is there to resist mechanical stress, like shear stress from two surfaces going like this. The adhesion belt is going to offer tensile strength to the top of the membrane. So for example, if you were to put shear stress like this across the top of an epithelial layer, you have these tight junctions. What's to prevent the plasma membrane, that apical surface of the epithelium, to be torn apart, right? To have that plasma membrane just torn apart. Well, it needs to resist that, and that's the job of the adhesion belt, right? It forms kind of like a stiff. It adds, not a stiff isn't the right word, but it's a strong sort of meshwork. The word is tensegrity that it applies to that surface on the top of the epithelial sheet. But good thought. So, and then the last are gap junctions. Gap junctions are different. The job of a gap junction is to create little pores in the plasma membranes of adjacent cells for the passage of small solutes back and forth. And so what this creates is, the term for it is synstitium. And that just means that the cytosol in one cell has the same osmolarity as the cell adjacent to it, and if there's something floating around in one, it's able to pass through the gap junctions into the next. So we sort of have this continuum of cells, all right, through these gap junctions. Gap junctions themselves are so fascinating, really. I again wish, I'm just like hinting at so many exciting things to talk about, but gap junctions, particularly in neurons in the brain, are responsible for, if you're into neuroscience, are there any neuroscience majors in here? A couple of you guys? Yeah, gap junctions, the function of gap junctions in neurons is an open area of investigation, but it helps share electrical field properties inside of cells and that adjacent cells that are connected with these gap junctions. And that has an enormous number of implications, which I won't get lost in the woods talking about now, but maybe someday over some cup of strong tea we can talk about it. Okay, I've already hit that, so let's. That was in lab yesterday. Okay. Questions? No. Glandular epithelium. So I've already said this. Endocrine glands are going to release their cellular components, their secretions, or I guess the word is hormone, because endocrine glands all make hormones. That hormone is going to get released into the interstitial fluid, which is then going to be absorbed into the bloodstream. So I said it went directly into the bloodstream. That was a simplification. It gets released into the interstitial fluid that surrounds a cell, and then that gets taken up into the bloodstream. There are no ducts in endocrine glands. And on the other hand, there are exocrine glands. So exocrine glands are ones that release their secretions into a duct of some sort. They're releasing it directly into some passageway that is going to convey that cellular product onto whatever surface that it's meant for. There are three types: merocrine, apicrine, and holocrons secretion. I'll go through what each of those means in a minute. But I just wanted to put the little definitions up there, that a secretion is something that your body uses, it's something that's useful, and an excretion is something it's trying to get rid of. All right, here we go. So there's this sort of continuum. And I haven't really thought about it very hard, but I've never really come up with an acronym or anything to help me remember which of these is what. But the order is MAH, Merocrine, Apicrine, Holocrine. And they form a continuum here. So let's look at that. Merocrine secretion, these are things that are produced in the Golgi, and they're getting released by vesicles, that is via exocytosis. So a common example of them is sweat glands. And this is just the sweat that your body makes to cool itself. All right? And if you think about that, you're just trying to release water and get the evaporative cooling from the heat of formation or the transition energy that is taken away from the surface of the cells as it goes from liquid to gas. So then the next is apicrine secretion, and it's a little bit more extreme. It's not exocytosis. What happens here is along that adhesion belt, the epithelial cell kind of pinches off and then sheds the whole apical portion of the cell itself. The whole cell gets released. So there'll be vesicles up in there that have their fluid components, but then like all this other stuff, the whole front end of the cell gets released into the duct. An example of that is a mammary gland. So a mammary gland that is producing mother's milk. Why would a mammary gland want to do this? Does this make any sense to you? What do you know about milk, and how can you rationalize this behavior? Yeah, what do you think? Okay, follow that idea. Mm-hmm. Okay, there's some stuff in the cytoplasm. Yeah, Walter? Huh? Well, okay, there's certainly going to be more calcium in this. Yeah, absolutely. It involves calcium. There's more to it. There's more to it. What thing do you know for certain? For certain, given your basic understanding of what a cell is made of and the different chemicals, biochemicals that make up a cell. Yeah.
You kind of need all of them. And so when you're pinching off these little pieces of cells, they can break down those pieces. It's more readily available, secreting tiny molecules. So you need all the macromolecules.
Yeah, and in particular, what proteins? What do we know about milk? What do you know about milk? What do humans make out of milk from a cow, for example? What do we do with cow milk? And I'm not talking the glow in the dark blue skim milk or something. I'm talking about like milk out of a cow. What can you make with it? Cheese, yeah. What is cheese? Is cheese mostly? Was that fat? Yeah, that's right. There's a ton of fat in baby's milk. There's a ton of fat in baby's milk. And what is fat? Where do we get fat? Fat is what we make. Phospholipids are what we make the plasma membrane out of. It's the ideal way to transfer fat. Okay, these lipids. It's the ideal way to transfer lipids because lipids are hydrophobic. You can't just dissolve them, right? So they have to be suspended. Milk is what's called a colloid, if you know that term. Any of you who've taken general chemistry, maybe. A colloid is going to be this suspension of large particles that stay up in solution. The fat, right? So this apicrine solution is used in mammary glands so that it can send, yes, Protein, yes. Calcium, yes. All of the other things that are involved in the cytoplasm, but specifically fat, because you can exocytose proteins. You can exocytose all kinds of stuff, but fat is hard to transfer. So they use apicrine secretion in the mammary gland. All right, holocrons secretion. This is like even more radical. This is like cells just blowing themselves up. They in apicrine secretion, you see it in stratified epithelium, where the cells get pushed eventually to the surface and then just burst. They just burst and release all their cytoplasmic content. So, for example, sebaceous glands. So that's the glands that are on your skin that make skin oil, right? And by blowing up, they release all of this oily wax onto the surface of your skin, make it water repellent, keep pathogens out, et cetera, et cetera. All right. So in kind of the same way the apicrine secretion, but we're going even further. We want to just like blanket the surface with a waxy hydrophobic coat. All right. So that's the whirlwind tour of epithelium onto connective tissue. All right, so there are three basic categories. There's connective tissue proper, these fluid connective tissues, which I talked about as being the blood and the immune system, the white blood cells, and then supportive connective tissue, which are bone and cartilage. Connective tissue binds organs together in terms of tendons and ligaments, or beyond that, though, reticular connective tissue can form the sheaths around various organs, like the kidneys or something. It acts to support your body, bones and cartilage. Physical protection, the cranium, the ribs, the sternum, of course, the bones. But also, like I just said, with the reticular tissue, the capsules that are around many organs that protect that organ. Well, movement, okay, sure. Bones act as levers. Storage, we've talked about how adipose tissue is a connective tissue and stores fat. Calcium can be stored in bone, and phosphorus can be stored in bone. Heat production. So brown fat in infants is an exceptional way to do a number of things, but one of them is to generate heat. Brown fat is how a teeny tiny baby is able to stay warm easily, and they're able to turn over that fat very rapidly. It's also an excellent way for them to get rapid access to the energy that they need to go through that explosive change, which is going from like a crying amoeba poop machine to like this running cute little terror that is suddenly running around everywhere. And then transport in the blood, and of course connective tissue forms the basal lamina throughout the body, all right, all over the place. So that's an overview. Let's look a little bit more closely at the connective tissue proper. So here's the proper, and it gets broken into loose and dense. I had gone through some of these yesterday in lab. And the loose, there's areolar, adipose, and reticular. I talked about areolar as being particularly important. The really stretchy stuff, like this underlying your skin, makes your skin stretch and come back to shape. And then there's dense connective tissue. The only one I really talked about there was dense regular connective tissue in terms of the tendons in your body. And then there are these specialized types. There's the supportive connective tissue, bone and cartilage, and then there's the fluid connective tissue, that is your blood. There are 9 general types of connective tissue proper cells. And there are a wide variety within each of these nine cell populations. I probably am going to skip through a few of these here, but I'll talk about a few of them here. I have them all in this slideshow. I might skip some as I go. These are important though. Fibroblasts and fibrocytes. Fibroblasts. So what did I say about words that have blast in the name? Huh? What did I say about? Remember, yesterday, what cells were we talking about? We're talking about osteoblasts. And what did I say about an osteoblast? What's that? They build bones. Osteoblasts build bones. Build. Yep. What else did I say about the word blast? You guys remember that? What else do osteoblasts do? They can mature, can't they? And they become osteocytes. It goes osteoprogenitor cell, osteoblast, osteocyte. And here, in connective tissue, you might think it would be fibroprogenitor cell, fibroblast, fibrocyte. And maybe you could use the term fibroprogenitor cell, but that's actually not the word that we use. Well, the word is mesenchymal cell, and we'll see that in a moment. It goes mesenchymal cell, fibroblast, fibrocyte. So fibroblasts are by far and away the most abundant connective tissue cell in the body. They're everywhere, tons of them. And the second most abundant are these fibrocytes. So fibroblasts are going to make all kinds of cellular products that act as sort of glue. They make a lot of high hyaluronic acid. I'm going to show you a picture of high hyaluronic acid in a little bit, and we'll talk about what that kind of intimidating sounding word means. But it's essentially just the glue that makes a connective tissue connect things. All right? And then fibrocytes. So the cellular products of a fibrocyte are a little bit different than that of a fibroblast. They tend to make more filamentous proteins, specifically one called fibrin. Fibrocytes make fibrin, which is this filamentous protein that, for example, is the structural component of clots. All right, but it adds strength to connective tissue in various contexts. So the fibrocytes are sort of like the maintenance vehicle for connective tissue. All right, so then I probably should switch these. Mesenchymal cells, these are the stem cells. I cut out, I don't know, did anyone look at the PowerPoint that I had posted for today? Yeah, I think I had in the one posted a slide about embryology, the embryonic development. I cut it out, but these mesenchymal cells are derived from the mesoderm, and they're basically the stem cells that are going to be sort of the grandfather or grandmother of all of the connective tissue cell lines, particularly fibroblasts and fibrocytes. All right. So, but you can get them to turn into macrophages, et cetera, et cetera. They're not embryonic stem cells. They are connective tissue stem cells. All right. So they're like as undifferentiated as a connective tissue cell is going to get, if that makes sense to you. And then adipocytes, fat cells. There's a lot more to say, but we won't. Macrophages. These things are so awesome. You guys are so lucky to live in the age that you live now with these like amazing microscopy techniques and like generations of scientists that have been spending their lives working on them, so that now you can watch a 30-second video if you go to YouTube about a macrophage that really looks like something out of a 1950s horror film that's going to come and eat Philadelphia. There are these amazing cells that are enormous, and they just send out these pseudopodia, and they crawl around, or some of them are fixed, they don't crawl, but they look for pathogens or anything that doesn't belong there, even if maybe it's your native cell. I forget the number, but something like over 1 million cells an hour in your body become cancerous and need to be eliminated by the immune system. And that's the job of the macrophage, is to recognize that and absorb those cells. You can see in this picture here, so here's a macrophage, and it's sending out all these sends out these arms that sort of project from the body of the cell, and then are surrounding these bacteria. Here's some sort of bacteria, some coliform bacteria that the macrophage is surrounding. So macro meaning large, phage meaning eat, a large eating cell. Mast cells. Yeah, we'll talk about mast cells some other time. Yeah, do I really want to talk about them? I don't think so. Lymphocytes. Again, these are blood cells. These are blood cells. They're important, but I'll talk about blood when we get to cardiovascular. I think I'd rather skip all these. We will talk about melanocytes a little bit today, maybe. They're a type of connective tissue cell that makes and stores brown pigment called melanin. This is what gives your skin its color. And you may think that a person with dark-colored skin has more melanocytes than a person with light-colored skin, but that's not true. All people have about the same density of melanocytes, and it's just a difference in the amount of melanin that melanocyte population is making. So that's really it. Okay. So those are the cells. Let's go through the fibers, the fiber types. These are the common fibers that we'll run into. Collagen fibers. So we saw them a little bit yesterday. This looks like some areolar tissue. This is a collagen fiber. Here's a collagen fiber. Here's another. Here's one. I also showed you a picture of collagen when I was talking about the piezoelectric effect. So they're very strong, and they resist force along the linear axis. Reticular fibers are also quite strong. However, reticular fibers are more like a spider web going in all directions. All right, so they are very strong and resist force in a three-dimensional orientation. Whereas elastic fibers, they're nice and stretchy. Sometimes you need things that stretch, and elastic fibers do that for us. Branched, wavy, you can stretch them. They're like rubber bands. They come back. Okay, so here is a picture of a cartoon of areolar tissue. Now, the reason I have this here is to show you, first of all, that, there's all these fibers, these cell products, and there's all these different cells. If you look at this field of view and then take all of the cells and remove them, what is left is called the matrix. And I'm not talking about Keanu Reeves here. I'm talking about the proteins, the filaments, the fibers, and the pink stuff in this drawing, this goo. That goo is what we call ground substance. It's not actually pink. It's clear. It's colorless, viscous. And it's there to fill the space between the cells and slow pathogen movement, right? And I think about it like imagining going swimming in a pool of Jello. What would that be like? Kind of fun, actually. I would try it. Why not? But probably not very easy, would it be? Yeah, probably not very easy. Or imagine somewhere between Jello and honey. That's kind of like sticky. I'm just thinking of food stuff. I don't know. Yeah, swimming through a pool full of honey would be kind of hard, wouldn't it? And that's what ground substance is there for. It's to prevent little bacteria from, say we get a scratch, and it gets past that cellularity layer of our cells in the epithelium. It gets into the connective tissue underneath. We don't want it running amok, just going any which way. We have to slow it down. It gets slowed down with the glycose, with the ground substance. This is made of a host of different proteins here. These classes of macromolecules, glycosaminoglycans or GAGs, proteoglycans, various adhesive glycoproteins. So let's see what that means here. First, the glycosaminoglycans. I talked about high hyaluronic acid just a moment ago when I was talking about fibroblasts and the proteins that they make. The glycosaminoglycans, there's a number of them. Chondroitin sulfate is the most common. You're going to find that in a lot of blood vessels, bones, and in articular cartilage. This is the chondroitin sulfate is the stuff that is responsible for cartilage being sort of stiff. And then there's hyaluronic acid. It's much more slippery and viscous than chondroitin sulfate. It's part of synovial fluid, which is the lubricant in a joint. It also forms the vitreous of the eyeball. So inside your eyeball, there's a clear jelly marble that helps your eyeball maintain its shape, and that has high hyaluronic acid in it. Let's look at these. I don't have. Oh wait, what do I have here? So here's high hyaluronic acid. Here's keratin sulfate. That's another one. Here's chondroitin sulfate. This is an example of a glycosaminoglycan. You're going to have this like filamentous protein here that has all of these long chains of carbohydrates coming off of them. All right? Carbohydrates are just chains of carbon with a lot of hydroxyl groups sticking off of them. But you'll notice, look at these different carbohydrates. I don't know how much experience too many of you have in the classroom with carbohydrates, but what can you tell me when you look at them? What looks a little bit different about those carbohydrates than other carbohydrates you might be familiar with, like glucose or sucrose or something like that? What can you see just looking at them? What looks sort of different about them? Chemically, what do you notice? Don't be shy. Take a chance. Yeah, you, yes. Remind me your name. Sally. Hey, Sally. Charges. That's it. That's right. You all saw it. She named it. There are these charges on here. Okay, these are acidic groups that at the pH that these guys find themselves at are actually in the conjugate base form, which is the anion, right? So you have the anion form of all these acidic groups. What happens when you put a bunch of negative charges near one another? What happens? Let's just make it a metaphor. What happens when you have two really negative people near each other? Do they become bosom pals? No, they repel one another. Negative charges repel one another. You guys have known this since third grade. Just shy. Okay, so here let's look at what this means a bit. Negative charges repel, these anionic repulsions. Another aspect of this is all of these hydroxyl groups are really good at hydrogen bonding to water. Much like, what are these things called? The Zorbs? No. Orbeez. Yeah, that's right. You put water in them, and look at them, they blow up. in the same way. They absorb water, right? So these glycosaminoglycans, these glycoproteins, their job is, first of all, to absorb and tightly hold water, to maintain hydration, all right? You want your cartilage to stay hydrated. You want your connective tissue to maintain its hydration state so it can be this viscous mess that the bacteria can't swim through. You want the vitreous of your eyeball, which doesn't have blood vessels going through it. You wouldn't be able to see through that. It needs to stay hydrated. So that's why we have these highly hydroxylated carbohydrate structures that are going to be hydrated and stay hydrated. And then all these negative charges on them are going to make it slippery. One of them is going to slip off the other one really easily because it's not going to be attracted to it. It's going to be repelled by it. So it's going to be a very slippery but viscous solution. All right? So I'm not, I would never expect you guys to be able to draw this stuff off the top of your head. Of course not. I mean, I have a PhD in carbohydrate chemistry, and I would have a hard time just whipping it out like that. But I do want you to understand how the chemical properties give it the structure, the chemical structure, gives rise to the function that is so important in these tissues. All right, that's what I want you to appreciate. Okay, so this is essentially the same slide I showed you the other day. I'm going to skip some of this stuff. I don't think this is so important. I've been going at length on other things here. I'm sorry, I should have snatched this stuff out. Yeah. This is an important concept, if I haven't already said this. So we said that epithelial tissue is avascular, right? Because of the cellularity. But the underlying connective tissue is not. It's not. And areolar tissue often forms the basement membrane in a lot of structures. You find it all over the place. And in all of these tissue membranes that I'm going to talk about in a little bit, it's typically areolar tissue. Areolar tissue is really conducive to the growth of capillaries, to vascularization, right? So it acts as the highway or the landscape through which the highways of capillaries will lay themselves out. That's an important concept to have. All right. So then, reticular tissue I don't think is so important either. I'm skipping over it just for sake of time. Adipose tissue, I'm going to talk about this one only because my daughter is on the slide. There's white fat. Everybody in this room, the fat that we have in our body is white fat. Whereas in this charming child here, she is made up of some white fat, but also a whole lot of brown fat in that belly. And she's right on the cusp of getting rid of it too. She was just about to walk right there. Brown fat is brown because it's much more vascularized, first of all. It has a lot more blood vessels. And it has, I don't know where it is in this list. Oh, here it is. A lot more mitochondria. There's a significantly higher concentration of mitochondria. And the idea being that brown fat, these babies can mobilize those energy reserves very rapidly. So they spend, 10 months not really doing much, drinking a lot of milk and pooping, learning how to eat avocados after 6 months or so. And then all of a sudden they start pulling themselves up and they learn how to stand. And once that happens, they need energy quick. They cannot eat enough energy to meet the energetic demands that their body is putting upon them. And so for the next year or so, as their musculoskeletal system develops, they start mobilizing this brown fat really rapidly, right? All right. So supportive connective tissue. So yeah, again, the point that I made here is that bone is highly vascular. We've looked at some of the structure of bone yesterday. This is the supportive connective tissue, right? And then cartilage has this gel-type ground substance, right? The matrix here was made of collagen and the other glycosaminoglycans that then gets calcified with the hydroxyapatite, whereas here it's not. It's not. This is hyaline cartilage. This is going to have a sort of gel-like matrix with a lot of the chondroitin sulfate in it for stiffness, but also hydration. It's avascular, no blood vessels. Again, to do the little plug for not just yoga, but all kinds of stretching. Cartilage, those cells still need nutrients, right? They still need fluid. They have metabolic turnover, but they don't have blood supply going into them. And so the way the cartilage achieves that is it's just like a sponge. I'm standing here, I'm bouncing around, and I'm compressing my cartilage. I'm squeezing the metabolites, the waste products out of the tissue. But then when I do some yoga and do a forward bend, stretch out my vertebra, that is like pulling it in the other direction and pulling fluid into that tissue. Everybody achieves that to a certain degree at night when you lay down, right? When you lay down and you relieve the compression on your cartilage, that allows fluid to be drawn into the cartilage. But you can, particularly for vertebral health of the discs in your back, it's really good to do passive inversions, so sort of a gentle traction to help that cartilage get the fluid it needs. In your joints, athletes, warming up a joint slowly, particularly as you age, but at all ages, particularly when you're like a high-level athlete, warming up a joint is so important. I remember I read everybody's slip yesterday, by the way. Thank you for those. They were some of them were very interesting. Learned someone works at, I forget who now, though. Someone told me they work at the animal shelter. Yeah, you. Okay, great. Thanks. My daughter, oops, she's not there anymore. She's collecting cans for the animal shelter. The point was, when you are an athlete, you want to warm a joint up. So the warmer synovial fluid in a joint can flood into that cartilage and help the cartilage swell a little bit to give you more cushioning protection when you move into the higher intensity phase of your workout or dance or whatever it happens to be that you do. So warm up is important. Warming up the joints you're going to use before exposing them to high impact is how you have long-term joint health. And I'm telling you folks, take it seriously. Take it seriously, because none of us in this room are getting younger. Although I feel like I'm getting younger sometimes. All right. Wait, one other point I wanted to make here. This is the point. Say I am stupid, and I have been stupid in the past. I was kind of stupid this weekend. Not really stupid, but I went downhill skiing. I break a bone, okay? I break a bone versus I'm skiing, right? So I break a bone and I do some weird thing with my knee. I tear my meniscus. I didn't. I might have done something with my shoulder. I don't know. But which of them would you rather have? A broken tibia or a torn meniscus in your knee, and why? He says tibia. What do you say? Bone. Okay, well, defend yourselves. What do you think?
The bone.
Okay. Yeah. Bone heals easier than cartilage. Bone heals easier than cartilage because it's vascular. Cartilage is avascular. This is why cartilage tears are such a drag. Be nice to your cartilage, your soft tissues. That's from experience. I was a wild youth. I definitely hurt myself. Okay, what do I want to talk about here? Well, this is kind of the same thing. So chondrocytes specifically produce these anti-angiogenesis factors. They produce proteins that inhibit blood vessels from growing into them. All right? Is there anything else new on this? Perichondrium is not so important, I think, to my purposes. I'll skip it. But, okay. Yeah, I should have cut these slides out. I'm sorry. I really am trying to focus myself, edit myself on the fly here. Okay. Yeah, let's get into some fluid connective tissue just a little bit. So there's two types. There's the erythrocytes. These are the red blood cells. And then there are the white blood cells. And there's a host of these. I had shown the pictures, but I flashed past them earlier. The lymphocytes. There's monocytes, lymphocytes, eosinophils, neutrophils, basophils. Not something you need to memorize now. I'll talk about them in lab, is what it'll be. I'll talk about them in lab. Yeah, let's keep going. This is where I want to be. This is what I want to talk about. Sorry, I flew past all that. Not important to my conversation here. So tissue membranes. I started the day by saying this is where I wanted to introduce epithelium and connective tissue so I could get you to these tissue membranes. There's 4 types of tissue membranes. There's mucous membranes, serous membranes, which we talked about yesterday in lab. Does anybody remember first of all, how many serous membranes are in the body? How many of them were there? James is thinking. He's counting them. He's got like 2. Like, what was the third one? Huh? There's 3. Yeah. No, I was just, yeah, there are 3. What are they? Do you guys remember? Yeah, James is here. The pleura, right on. The peritoneum, number 2. The pericardium, James wins a cookie. Invisible cookie, though. The better for you. That's right. Those are the 3 serous membranes. Synovial membranes make up the joint cavities, the joint capsules. Mucous membranes, I'm sorry, I should have just skipped over them. Mucous membranes they line the surface of cavities open to the world, right? So they're inside of your nasal passages, your esophagus and throat, your pharynx, in your urethra, the vagina, passages that make mucus that are open to the outside world that need to stay hydrated. That's a mucous membrane. Serous membrane covers surfaces that are not open to the outside world. They're enclosed to the inside. One thing I didn't really go into with serous membranes yesterday, and maybe I'll mention now, because I don't know if I have another slide that talks about them in here, is why does the heart, why do the viscera, the abdominal organs, and why do the lungs have these 2 surfaces, the parietal and visceral layers of those serous membranes that have the like slippery exudate that they make on the inside of that cavity between those 2 surfaces? Why do they have that? What do those 3 things have in common besides being in the body? Yeah. That's right. Yeah, your heart is constantly moving. Your lungs are constantly inflating and deflating. Stuff is moving through your guts, right? And so we don't want friction, right? Like how horrible every time your heart beat, it was like, So that's what serous membranes synovial membranes or joint capsules. And then lastly is the cutaneous membrane or skin. The big thing to remember here is that one of these tissue membranes is epithelial tissue over a layer of connective tissue. That's a tissue membrane. That's it. ET over CT. Epithelial tissue over connective tissue. That's the definition of a tissue membrane. All right? The grandest takeaway. So yeah, here are the mucosa. They're lining passages that have external openings in the digestive, respiratory, reproductive tracts. Yeah, I guess I had already said all this stuff. But the connective tissue that is the underlying the epithelium is called the lamina propria. That's the term that you'll see in medical textbooks and how it's referred to. So this areolar tissue is called the lamina propria. The proper layer, lamina layer, propria proper, the proper layer of connective tissue, the epithelium is lying on top of. So this is the mucosa, and you'll see in the mucosa there's these columnar epithelial cells, and then sometimes there'll be these goblet cells. These goblet cells are going to make the actual mucus that will get released onto the surface. So the mucus is this like sticky, we all know what mucus is. We've all experienced it. And it serves to catch particles that maybe you inhale in your nasal passages or whatever, and you don't want to make it all the way down into your lungs, get stuck to the surface of the mucosa, and then the cilia kind of bring it back out to the mouth or whatever. All right. Serous membranes. Yep. So I guess we'd already talked about this. Fluid transudate reduces friction, like I said. Again, I guess this is concepts I've already covered. Yeah. In the serous membranes, we call it a transudate, the name of the fluid that is produced by these squamous epithelial cells that are producing the fluid that's going on their surface, the slippery stuff. When we dissect the pig hearts, what is it, next week maybe? And we get the pig hearts, most of them will have the visceral pericardium. Some of them will actually still have pieces of parietal pericardium, and you can dissect that away and be able to identify this connective tissue sheet that lays between the myocardium, the muscle wall of the cell, and the parietal pericardium. All right. So said this. James already reminded us. That was impressive. Synovial membranes. So they're lining these moving, articulating joints filled with synovial fluid. One thing about the epithelium in synovial joints is it's often incomplete. So it's not often it's not a complete layer of epithelium inside these synovial joints. But they produce this synovial fluid, which can get absorbed and nourishes the chondrocytes in the hyaline cartilage of the articulating surfaces. Because it's an incomplete epithelium, they say it's not a true epithelium. But there are epithelial cells there for sure. All right, and then lastly is the cutaneous membrane. This is what I'm going to spend the rest of the time talking about today. So the skin is covering the entire surface of our body. It's thick, it's waterproof, and it's dry. All right, here is an intimidating looking outline of all of the material I used to cover I've learned to talk a little bit slower and cut stuff out. But this is what I used to cover when I talked about skin. I won't talk about all of this for sure. I won't talk about hair and nails at all. I think I blacked out the skin cancer here today before I started talking. But I will talk about the epidermis and the dermis. There's essentially three general layers. There's the epidermis, which is the epithelial cells, the dermis, which is the connective tissue layer of this tissue membrane, and then there's the hypodermis, which is the fat layer that we saw yesterday. Isa? No, you're not Isa. No. It starts with an A. Artricia, yeah, thank you. There's an Isa here somewhere. Oh, there you are. Okay. All right. So, what? 15%? Holy smokes. That's a lot. 15% of the body weight is your skin. It's pretty amazing to me, but it's only 2 square meters in area, not as much as you might think. And only about 1 to 2 millimeters thick, depending upon where in the body we're talking. 1 to 2 millimeters, 15% of the body weight. That's a lot. So it's made up of course, the cutaneous membrane, which is your skin that has the epidermis and dermis. I guess I could put hypodermis in there. That's the fat and fascia that's underneath all of it. And then there's all these accessory structures like the hair and the sebaceous glands and stuff that I'm probably not going to talk about at all. All right, so let's go here. Within skin itself, there are 2 types of skin, and I think we're probably familiar with that. There is thin skin and thick skin. Thin skin has 4 layers of keratinocytes in the epidermis, and thick skin has 5 layers of cells in the epidermis. And thick skin is called hairless skin because there's no hair that grows in them. Like on the palms of your hands, on the soles of your feet, on the ends of external genitalia is a little bit more, and your lips, it's a little bit of this. So you're thinking, and then of course this skin here it's got hair on it. You may be thinking, wow, that's some pretty thick skin back there that this gentleman has, and these baby's feet seem so thin. It's not about the actual thickness, right? Of course, the actual thickness of anybody's skin on their back is quite thick, right, compared to that of your hands. But it's about the number of layers of cells, the cell morphology and the cell identity as it's going through the layers of the epidermis. All right, so believe it or not, I made this slide prior to 2016. Functions of the skin. Well, one of the main functions of the skin is to convey facial expression. So as I stand up here talking to you, I sort of read people's facial expression. If you're looking at me like that, then I know that you don't understand what I'm saying. If you're like this, I know that you don't care what I'm saying. We all read facial expressions quite well, and that is through manipulations of the skin, as obvious as that may seem. But it has a lot of other functions. So it's protecting, it provides a thermal buffer, it protects us from UV light, it acts as a water barrier, mechanical and chemical protection, absorption of whatever can come through the skin. It forms a barrier to pathogens. Macrophages live in the skin that destroy pathogens that think they have a chance. I guess hot-cold protection is the same as, it's not the same as thermal regulation. By thermal regulation, I mean the capillaries in the skin help us radiate heat off, is what that means. and then we derive sensation through the skin. Importantly, so I kind of skipped in the sake of time yesterday, going into depth about how vitamin D really works and the story of vitamin D, although it's a pretty simple story. We get about anywhere from, I don't care how much milk you drink, 80 to 100% of your vitamin D that your body needs for calcium regulation, calcium absorption in the gut is actually coming from your exposure to the sun, right? This is why people that live in northerly climes where it's overcast in the winter, like in the plain states of Canada, you know, the provinces of Saskatoon or something like that, those people up there, they can have vitamin D deficiencies. You know, rickets. I didn't talk about rickets yesterday, but you got to get a little bit of sun, a little bit of sunlight in measured doses. Yeah. So, epidermis. The epidermis is avascular. I've already covered that concept. But nutrients and oxygen that the epidermis needs diffuses from the underlying connective tissue. All right, so whatever nutrients and oxygen that the epidermis needs are coming from below, come up and diffuse from the capillaries in the dermis. We live in the Me Too era. Sorry, James Bond. I put this picture in here because this, I don't know if anybody's ever watched this movie. I'm not necessarily suggesting it. It's called Goldfinger. It's one of the James Bond movies from back in the day. There was this the scene in there where the heroine or whoever that was, the woman, she dies because she has been dipped in gold and suffocated. Because at the time, this was just in like the late 60s, I guess, when that movie was filmed. They thought that you absorbed a significant amount of oxygen through your skin. And the idea was that if you covered your skin completely in paint or gold, I guess, then the person would suffocate to death. Of course, that's absurd. Of course, that's absurd. That is not how your body your body breathes. You don't breathe through your skin. But this was so legitimately what they thought that in this actress, she's wearing, thankfully, some briefs there. And under those briefs, there was no paint because they were worried about her actually suffocating on the set of that show. Of course, that none of that happened. And we have a different understanding. But there's a weird story for you about James Bond. All right. Here it is, the skin. Let's dissect the epidermis, if you will. So this is an example of thick skin. I have all 5 of the layers here. This bottom layer, the very first layer, the most important for you guys to really know about is called the Stratum Basale. It's also it also goes by the name Stratum Germinativum. I think I have that on a different slide. I should probably put that there as well. I prefer the term Stratum Germinativum. That's how I learned it, and it makes sense to me because it's the germinative layer, the layer that gives rise to the stem cells. Also, I guess maybe the basal layer. But this is the layer of the epidermis that has the stem cells. That these are the cells that are going to be in close contact with the capillaries that are going to be the most nutrified, the most metabolically active, have the most access to oxygen. Then as we move up through the layers, these cells continue to divide and push their sisters upwards towards the light. As they get up there, these cells in the middle get further and further away from the source of oxygen and nutrients, and they go through this process of becoming quiescent and then eventually dying. They eventually die, and as they die, they release all of their cell contents, and form this sort of dead, waxy barrier at the very top of the skin. I've starred this one here, stratum lucidum. Stratum lucidum is the extra layer in thick skin that is not present in thin skin. All right, so thin skin would have these four, and thick would also have the stratum. The only one that is really important, I think, for you to understand is this idea of stratum basale, the germinative layer where the stem cells. Some other things to point out here are some melanocytes, and they live in that stratum basale, and they send out arms that are pretty magically, they have proteins on them that are magically able to penetrate through tight junctions, and they deposit little bits, little vesicles of melanin into the cytoplasm of all the other fibrocytes. All of these cells are types of fibers. This is all fibrocytes here. But this has melanin, these melanocytes, the way that they work has implications in terms of melanoma, which is a type of skin cancer. Melanoma is particularly aggressive because it is able, it's coming from a cell line that inherently is good at being metastatic. It's inherently good at invading tissue. All right, so melanoma is definitely not one to screw around with. What else do I want to point out in here? I think that is pretty good. Yeah. So here is sort of the life history. It takes about a month to go from the bottom to the top. And these keratinocytes that come from the stem cells in the stratum basale, they move up into this large stratum spinosum that's right here. A stratum spinosum is so called because it's these like spiny-looking cells that have cytoskeletal elements that make them look kind of pokey. And then finally, we're getting up towards the surface. When we get into stratum granulosum, these cells have a lot of granules inside them. They're speckled-looking cells that are full of oil and waxy products that are, when they compact down and they start to die, they form this layer over the top of the cell. And then finally, when we get to stratum corneum, these are just the dead skin flakes. They're desiccated, a dead skin flake layer. Cornea, is just something that it means clear, right? So it's this clear layer of dead skin cells that's on the very surface. It takes about a month to make its way. All right, on to the dermis. So dermis is made up of two layers. The papillary layer is just this little layer beneath the epithelium that has these papilla, these little bumps, these hills, papillary layer, and then the reticular layer is just below it. Papillary layer is made up of areolar connective tissue, and then the reticular layer is made up of what's called dense irregular connective tissue. It has a lot of reticular fibers in it. That reticular fibers, you'll remember, are those fibers that are strong in 3 dimensions all around. So the dermis is both stretchy because of the papillary layer and strong because of the reticular layer. Then below that, so this dermis is where all of these accessory structures more or less live, like Pacinian corpuscles, we'll talk about them maybe next week, sweat glands, et cetera, hair follicles. Below the dermis is all of this stuff down here. That is the hypodermis. This is really where the fat lives, the thermal barrier. So when you're getting an injection, you can either get a hypodermic injection. You're probably familiar with that term, where you're going through the dermis and down here into these larger arterioles, or you can get a transdermal injection that's just in the dermis that is putting the tissue, and it's going to be a slower release because it's got to work its way through these capillaries down into the larger blood flow. Yeah, everybody cool? This is all almost pretty much just taxonomic here. All right. Oh yeah, we talked about this. I just talked about this. Subcutaneous fat, that's below the dermis. Let's keep going. Skin color. So this is what I was talking about in terms of the melanocyte. Here's a melanocyte, and you can see that these arms that the melanocyte is putting out are able to travel between the layers of the epithelial cells. And then little bubbles get pinched off of the end of those arms, get absorbed by the cells, and that melanin gets incorporated. You can, and I think you can begin to appreciate from the way this drawing is depicted how just a few gene changes in this cell, which is so open to UV assault from the sun, can lead to a real problematic situation. See what I have time for. If I have time, I'll show you. I crossed it out, but I have a slide that talks a little bit more about melanoma. Yeah, so I guess I had said this already. I guess I had assumed I had eliminated this. But people with dark skin are just producing greater quantities of melanin than people with light skin. And this protects these people from the advent of UV radiation damage to their skin. Yeah, and the melanin that's produced is also turned over much more slowly in people with darker skin, conferring that advantage. And so I guess that's all I have to say there. I really don't need to talk about all this. There's a bunch of different things. Okay, yeah, let's talk about this. I'll give you a little bit of pathology, and then I'll let us out just a bit early today. So here's a story I'm going to tell you guys about a camp I used to work in. I used to work at a camp for children with chronic skin disease. And I have atopic dermatitis, and I sort of wanted to work at a camp through the American Academy of Dermatology. It was an amazing experience getting to work with these young people. And there were kids who had atopic dermatitis, very acute, like myself, and many other skin disorders, ran into a lot of them. But by far, the most arresting was my exposure to these people, people with RDEB, recessive dystrophic epidermolysis bullosa. So what happens here is, and I want to talk about this a little bit because I think it's instructive in terms of understanding the skin. The collagen 7 gene. There's your body has, I don't know, like 20 some genes encoding different collagens depending upon the context in the body that the collagen is found in. The collagen that you find in the dermis of the skin is collagen 7. And there can be a number of different types of mutations that happen in this gene that can lead to a number of different types of epidermolysis bullosa. There's junctional EB, which is also horrible. There's RDEB. There's I forget the other names. There's about 3 or 4 or 5 different types of EB. Some of them are actually quite mild, certainly in comparison to this. These people with RDEB, they have virtually non-functional collagen in their skin. And so what happens to these people, who here has had a blister before? I imagine most of you guys have had a blister. When you get a blister, what's that like? It's it's a bubble, right? And it feels hard, right? There's like the fluid pressure that makes that blister like a kind of a stiff bubble. Hey, well, in these people, it's really easy to raise a blister in them. Incredibly easy, because any kind of shear force, like the gentlest shear force on their skin, will separate that papillary layer from the reticular layer of their dermis. All right, and they get a blister. However, in these people, the fluid pressure of that blister, the margins of the blister are not able to withstand that fluid pressure. So the fluid pressure makes the blisters grow. The blisters grow, and they can cover huge portions of their body, right? Huge, 80% sometimes of these people's body are either covered in blisters, or one massive blister, or are healing from a blister. And it goes beyond that. So it's not just on the surface of their skin. It happens on the into the connective tissue that's running through their GI tract. So these people have to be on liquid diets their entire life. And they have, you can see this mitten phenomenon here, where if you were to see this child's hands move, you would see that the fingers are inside those mittens there, but it's just been scarred over. These babies come out looking relatively normal, but with time, the effects of the disease take over. They have to, often they're really prone to corneal abrasion. They have in many ways, horrifying existence. But I think there is something really important in not being afraid of the experience that these people have, of not turning away from it. Because, I put these pictures up here, and I could put many more that are just gut-wrenching, just make your heart cry. And I've shed many tears about these situations. But it's important because some of the bravest people I've ever met have been people with recessive dystrophic epidermolysis bullosa, and they have things to give to the rest of us that only they can give. Messages of bravery and understanding and humanity that I think it's hard to find in the scale and scope that these people are rendering it. So I'll tell a specific story about a boy named Justin Cross. He had recessive dystrophic epidermolysis bullosa and was, at the time of the story, he was 12 years old, had blue eyes, beautiful blue eyes, young boy, and I was at this camp in Minnesota, Whitefish Lake in Minnesota, and I was doing my first bandage change on him. So these people have to go through bandage changes on a daily basis. So at home, normally 2 times a day, but at camp it was like once a day because it was like a major undertaking to do. And what you have to do is you take these people and they soak in the bathtub for quite a while, because even Telfa nonstick bandages are still going to stick to stuff like this. And then you slowly have to unwrap the bandages from their bodies, And as you're doing it, it's like, like a really, to most people, it would be such a humiliating thing if they had to undergo this. Like these people have to wear diapers often because they're eating liquid food, and because they've never had solid food, they've lost all the tonus in their rectum and anus, and so they have very little control over their bowel movements. And so you're changing a diaper that where they have sores on their backside that are in this in their waist. You're cleaning them up, and I'm unwrapping the bandages. I am scared shitless. I am so afraid of what I'm doing because I can see that everything I'm doing is hurting the child. It's clear that he was in pain as I was changing his bandages, and he at a certain point he stopped me and he said, "Tom," his name was Justin. He said, "Tom, don't be afraid. Do not be afraid. It's okay. I know it's going to hurt. I know it's going to hurt, and I'm not afraid of it at all. It's all right. You can do it." And he started giving me all of this encouragement, and just the ability of a person whose daily existence is so marked by pain, to be able to offer me, an individual with every advantage, the support and encouragement and comfort, comforting my humanity. That was just the most radical moment to me. It was the most radical moment. And so I bucked up and finished the bandage change on Justin, and then went out. It had been raining, and the rest of the camp was in the cafeteria listening to some guy with the guitar. And but the rain was clearing, and the camp is on a bluff, and I go out there. There's this picnic bench. I know this part's going to sound hardly credible, but it's true. I sit out there, and I'm watching this. The sun had, the clouds have cleared, and there's rain clouds misting back somewhere else, and this amazing rainbow, full arc rainbow opens up in the sky, as I'm having these revelations about the experience that I've just had. And then the whole camp trundles out and surrounds me out on the bluff because the rainbow's there, and the guy starts singing the Rainbow Connection. It was a pretty surreal moment for me. The point is simply that, we often would label these people handicapped, right? Handicapped. But I question the mindset that gives us, as if they have something less. I say it's something different, because that boy shared something with me that I don't know anyone else that I've ever met that could give me what he gave me that day, right? And so that is there, that is real, that is something that not every person with RDB is going to be that way. I met ones that were bitter. I met ones that were addicted to morphine because they're in constant pain. I had all kinds of things, but there is this potential. I've met so many of them that are truly amazing and inspirational. Here's one. This is a woman named Jamie Hartley. She was a counselor at this camp, and Jamie passed away sadly 3 years ago. She's been gone, but Jamie was the oldest living person in the world with recessive dystrophic epidermolysis bullosa. She made it well into her 30s with it, and often people make fun of the Mormon community for whatever reason. I never find any of that humorous because I've really seen firsthand how caring that community can be. She was able to live that long because she had just remarkable support from her family, from her community, and eventually from her husband, who married her. And dedicated his life to helping her live out her dreams. She was an exquisite singer. She had a very beautiful voice and got to perform in the Mormon Tabernacle, et cetera, et cetera. Just a lovely human being that I was very blessed to know. But yeah, she was another one of those sorts of people who had this thing to communicate. So that's my story at the end of the day here. I don't have much else to say, but thank you for listening to it. We're going to take a break now, and we'll start with lab in a little bit.